Thank you very much, and it's a pleasure, as always, to be at OHBM. As Hartley said, my group in Oxford are interested in using a number of techniques to understand the neurophysiology of motor plasticity. So, what is motor plasticity? For the purposes of this talk, motor plasticity, I'm meaning to refer any change in the brain that happens as a response to learning new motor skills and responding to new challenges. Okay? And that's important because I'm trying to encompass, and our group work really is interested in encompassing, both changes at a synapse all the way up to the changes at functional connectivity in functional networks that many of you may be more familiar with. We've had a very nice introduction to TDCS from Axel, but we've been interested in TDCS for a long time now because anodal TDCS in the motor system is thought to induce long-term potentiation-like changes. And that's important for us because a number of studies reasonably consistently have shown that when you apply anodal TDCS in the context of sequence learning, it improves healthy control's ability to learn a sequence. It improves our ability to acquire motor skills. And perhaps most importantly for our work, it seems to improve our ability to relearn and reacquire those skills after an injury, such as a stroke. So to illustrate that, I'm just going to talk you through very quickly through a small but randomized double-blind placebo-controlled trial we performed in Oxford a couple of years ago now, where we took patients who had subcortical lesions and were in the chronic stages of recovery after a stroke, and we gave them 9 consecutive days of motor training, the first 20 minutes of which were either with anodal stimulation to the ipsilesional primary motor cortex or a sham control. On the y-axis, you can see the ARAT score, which is just a clinical outcome measure where higher numbers reflect better outcome. The two groups are shown, anodal group in green and the sham group in blue, and you can see at baseline their scores were pretty similar, as we might hope. After the 9 days of intervention, both groups showed a significant clinical improvement, but the anodal group was over and above what we saw in the sham. And very importantly, that improvement lasted for at least 3 months, and anecdotally much longer in these patients. For those of you who aren't familiar with the ARAT score, and that may well be most of you, that kind of improvement is reflecting, talking to the patients is reflecting things like being able to peel a banana for the first time, being able to cut up a steak. Okay? So small, but behaviourally and functionally relevant improvements. However, and anyone who does know anything about TDCS or brain stimulation in general will know that the effects are massively variable. Some people respond very, very well to this kind of intervention, some don't. Now, there are 2 ways that we could start looking at that. One is to say, that means the technique doesn't work. Or to see it in the context of any kind of intervention and say, with anything we try and do in these complex situations, some people will respond better than others. And the important thing really is trying to work out why, so we can optimize therapy, and hopefully trying to predict who might respond and who doesn't, so that we can use this technique for people who may respond and something else perhaps for others. So along that line, as well as looking at the clinical outcome, which was the thing that we were most interested in, we also did some fMRI before and after. And what I'm going to show you is motor-related activation. So we've got them we passively moved their stroke-affected hand. And this is after the intervention compared with before, compared with the sham group. And what you can see is a significant increase in activity within the ipsilesional primary motor cortex after anodal TDCS compared with control. And if we look on a subject-by-subject basis, the degree to which people are able to increase that activity as a response to stimulation and training is reasonably closely related to how much they responded behaviourally. So it looks like this increase in activity in the ipsilesional M1 may be telling us something about the mechanisms of TDCS. And if we can understand that, we can start to optimize the therapy. So how can we break down the fMRI into things that tell us more about the neurophysiology? Well, the first thing we'd like to do probably is go back to the animal literature and see what's going on in the primary motor cortex when animals learn or recover after a stroke, or we induce LTP like plasticity in M1. And there are a number of ways that I could demonstrate this next point, but in the interest of time, this is one of the seminal papers that really tells us something about the physiology of plasticity in motor cortex. So this is a very standard paper looking at inducing physiological synaptic plasticity in the freely moving awake rat. And what they did was induce very standard LTP at the dotted line and looked at the response. And as you would imagine, for a given input to the presynaptic cell, the postsynaptic cell, after inducing LTP, gives us a very nice increase in signal, a classic LTP induction. However, if you do exactly the same thing, but in the presence of a drug that increases GABA, that increases the level of inhibition, you get absolutely no response at all. Okay, and this has been replicated in a variety of ways across a variety of studies. So this leads us to suggest and make the hypothesis that perhaps a decrease in inhibition is necessary for glutamatergic LTP to occur. And therefore, perhaps the degree to which we can decrease that GABA initially will tell us something about how much plasticity someone is able to mount. So can we see similar things in humans? Well, we can't do the kind of beautiful electrophysiological recordings that I've shown you, but what we can do is use a technique called MR spectroscopy, which I imagine many of you are familiar with. If you're not, it's a very theoretically simple, technically difficult way of using the standard MR scanners to get a measure of neurotransmitter concentration. So what we do routinely is select a region of tissue, always for us in the primary motor cortex, and that's a 2 centimetre by 2 centimetre by 2 centimetre voxel, so it's quite large. And we excite that region as a function, excite that region in the scanner, and then plot the signal coming back as a function of its frequency. And we see a very characteristic spectrum. This is a megapress sequence. And because we understand the structure of the molecules, we're able to identify a peak that represents the concentration of GABA within that region, and another peak that represents the concentration either of glutamate specifically or glutamate and glutamine together, depending on a number of technical factors. So this gives us a way of quantifying inhibition physiologically within our region of tissue. So the first question might be then, okay, so we've got a measure of inhibition. If we believe that a decrease in GABA is necessary for plasticity to occur, then if we do something in healthy controls that induces plasticity, like learning a motor task, do we see a decrease in inhibition? This has been shown before coming out of FIMRA. This is a replication by Emily Hinson and James Kosinski, my group. And what they showed was that if you get people to learn a motor sequence in pink, you do indeed see a decrease in inhibition over that period of learning. Whereas if they're just in the scanner moving their fingers or lying at rest, there's no change in inhibition at all. Okay. So I started off by saying anodal TDCS seems to induce LTP-like plasticity. And I've suggested that plasticity in M1 may be related to levels of inhibition. So can we go on and say, does anodal TDCS lead to a decrease in GABA as well? So a few years ago now, we did exactly that. And what we saw was, again, a very similar decrease in inhibition due to anodal stimulation in the stimulated region of about 10% after 10 minutes of 1 milliamp TDCS. And we saw no change with the sham. But that still doesn't actually show you that GABA is important in motor learning, and it still doesn't give us a potential mechanism by which TDCS may improve plasticity. So can we go on, rather than just saying both of these things induce this change in inhibition, it must be that thing that allows TDCS to improve plasticity. Can I go on and show you some data that might persuade you that actually inhibition levels are at least a key mechanism by which TDCS might influence our ability to learn? So this is a study that we did a few years ago now, where we looked again at anadal TDCS effect on GABA. So we looked at GABA levels in M1 before and after anadal TDCS to M1. We had a regional control. And on a separate day, we asked the same participants to perform a sequence learning task during fMRI. And the first question we wanted to ask was, if the decrease in GABA is an important mechanism in common between TDCS and learning, then the responsiveness of the GABAergic system to TDCS should predict how much people learn. So if you're good at dropping your GABA in response to TDCS, you should be good at dropping your GABA and hence learning on a separate day. So we got a measure of learning from an explicit sequence learning task, which was really very simple and was just the decrease in reaction time on a subject-by-subject basis as they learnt the task. And so what I'm going to show you is the relationship between the change in GABA due to TDCS on one day and the change in reaction times as a proxy for learning on another day. And we see exactly what you might predict, which is that people who learn more, so have a greater potential for plasticity, at least in this context, are people who are able to decrease their inhibition more in response to anadal TDCS. So this gives us perhaps a mechanism and a reason to explain at least some of the variability that we see in our brain stimulation. But the second half of what I was particularly interested in, and what I sort of set this up to be, is not only can we explain that variability, but can we use measures taken before a stimulation intervention to predict who might respond and who might not. So, okay, so what we did, in a previous study, we had used anadal TDCS to the ipsilesional M1 in a group of chronic stroke patients. And these are our results. And we showed that anadal TDCS decreased reaction times compared with the sham control. And we did that in two separate groups. And that's a reasonably robust finding. So anadal TDCS to ipsilesional M1 improves function on a single session. But we wanted to take it a bit further, and again, these results were very variable. If GABA is important in the mechanism by which TDCS improves learning, then perhaps GABA levels at baseline may predict who will respond and who won't. So this is work done in collaboration with Jacinta O'Shea in Oxford, and what she did was take that reaction time data and look on a subject-by-subject basis whether some spectroscopy measures of GABA acquired before we did the brain stimulation predicted the response. And it was a small number of subjects, but it was a reasonably strong relationship, such that the more GABA you have as a chronic stroke patient before we do anything to you, the more you're able to respond and improve in response to TDCS. So again, perhaps getting closer to suggesting that amongst the many reasons and the many mechanisms that TDCS may affect plasticity, perhaps a change in inhibition is an important one by which we can not only begin to understand variability, but also predict outcome. But I said at the beginning that we are interested in plasticity as a whole. I'm interested in the synaptic changes, and I've talked a lot about GABA. But we are here because we are systems neuroscientists, and we're interested in the whole brain. We're interested in functional networks and how brain regions may change their communication with each other to promote plasticity. And what I've shown you are a lot of results from a single voxel within the wider motor network, and use that to explain reasonably complicated changes in behaviour that are certainly not entirely driven by that region. So one of the things we've been working on for the last few years are to try and look at a more network level at neurotransmitter changes than just looking at a single region. So a few years ago, Amir, when he was in Oxford, developed a bilateral motor cortical approach. So this was aiming us to look at the neurotransmitters at both motor cortices simultaneously. If we look at the stimulated M1, again, we see exactly the same result. We see this decrease in inhibition, decrease in GABA with anadal stimulation. But if we look at the unstimulated one, we see a very, very similar result, actually, in a decrease in inhibition. Interestingly, if we just take those two changes on a subject-by-subject level, they're not correlated. So I don't think this is just due to current spread, but rather, if we look at the FA within the tracts connecting the two M1s, that quite nicely, at least in a small number of subjects, trends towards explaining that kind of the relationship between the two changes for both anadal and cathodal stimulation. But again, that's not really a network. And so we've been working again with Uze and Adam Steele, who's here, looking at whole network level GABA. The ability to look at glutamate in 9 minutes in this kind of resolution has been published recently. Adam has work on a poster here looking at GABA levels within 9 minutes and predicting various aspects of behavior with those. So if you're interested in this kind of work, please go and see the posters. So I'm going to finish, my time is up, I'm going to finish by thanking my group and collaborators in Oxford and elsewhere. If you are interested, please go and see the posters. I've given you a little taster of what they might be, but go and see them and talk to the guys that did the work. And if you're really interested, come and work with us. Thank you very much.